Okay, thanks, Bertrand and Roland, for inviting me to present the strategy that we are going to follow in the use case 3, G cross E cereals, to use phenotyping, to apply phenotyping assisted by models and from using multi-scale observations. So here, okay, here are the 6 institutions that contribute to the use case. So here are the list of the contact points from each institutions for each institutions. But for sure, there is much more people involved in the work. So okay, as you know, phenotyping is characterizing individuals by observable traits. So a phenotype, measuring a phenotype, is the goal of phenotyping, and the phenotype is the consequence of the interactions between genotype and environment. So if we can phenotype many genotypes in many environments, we can try to describe these interactions and to predict a given phenotype predict the phenotype from a genotype and a given environment. So when Roland and Bertrand ask us about the use case dreams in the kickoff meeting, we say, okay, to not make short, our use case dream would consist on observing like 1,000 of genotypes over 1,000 of environments, and then use crop process-based models to interpret these high throughput phenotyping observations and estimate what we call functional traits that are varietal parameters as described by the model that could allow us, okay, to generate gene to phenotype models and describe G cross A analysis, but also would be interesting for different applications like breeding, the evaluation of cultivars in the registration phase, but also crop management. Okay, for this letter to Santa Claus, let's say that thanks to the recent development in high throughput phenotyping platforms, the number of genotypes is less of a constraint, because thanks to these new instruments, we can monitor effectively a large number of plots, which means a relatively large number of phenotypes. In this case, in our J cross E use case, we selected, however, a panel of 40 elite cultivars where we know that there is a priori some differences on what concerns precocity, call requirements, the architecture, so basically the height and also leaf inclination, and registration date. We tried to choose new cultivars and some old ones, and also cultivars coming from different countries. So okay, we come back to the number of genotypes is not a problem, and in my talk today, I would like to talk to you about 3 factors. So the large number of environments, how to get a large number of environments, how to get seamless data sets, so data sets that are intercomparable regardless of the sites, and then how we expect to use models in order to estimate these functional traits. So increasing the number of environments. So down on the slide, you have high throughput phenotyping platforms in control conditions and also in field conditions, which are basically the most, let's say, the most popular approach today for phenotyping. Of course, in our use case, we incorporate these 2 scales of phenotyping with trials in control conditions in Montpellier and also in field conditions in France, in Portugal, and also in Switzerland. But, okay, based only on high throughput phenotyping platforms, we have a limited number of environments. We can increase the number of treatments per site to increase the environment. But okay, anyway, the environmental factors that we can play with is somehow limited, and also it increased geometrically the size of the trial. Also, we can increase the number of sites, but if we rely only on high throughput phenotyping platforms, the number of research infrastructure that have such kind of instruments is still relatively low. So in our use case, what we thought is proposing a multi-scale approach where we use different two types of trials where we are going to monitor the genotypes with Earth observation, with satellites. A third scale, which is satellite on research infrastructure, where we monitor the whole panel with no treatments, and another one that could allow us to monitor these cultivars on farmer fields, where we try on each farmer field, we monitor only 6 cultivars per site. So Earth observation is a tool that is, well, it has been used since many decades for the monitoring of vegetation in general, but until now it has not been really exploited for phenotyping. So one of the questions that we would like to reply in our use case with the support of the Work Package 3 is, can we phenotype from satellite? How to phenotype from satellites? In the remote sensing domain, there are what are called radiative transfer models, which are physical models that allows us to estimate essential traits like the green area index, like the fraction of absorbed photosynthetically active radiation, but also different traits related with leaf biochemical content, like the leaf and the chlorophyll, leaf and the canopy chlorophyll content. So the idea, this kind of radiative transfer models are general for all kinds of vegetation. And most of the time, what we do is in the inversion of this radiative transfer model, constrain this inversion to increase the accuracy over a particular canopy architecture. For example, here you have few works that have attempted to create specific algorithms based on radiative transfer model to estimate green area index of specific crops. So the results is for instance, like that. This is generated from a very high resolution satellite, PlanetScope. So these are maps at 3-meter resolution of the green area index. This is the result of the work of Rihanna McAneny in the National FAST project, where we can see in farmer fields 4 different genotypes. These are images that in the post-anthesis is that in grain feeding, and we can clearly identify how there are 2 cultivars, Fructidor and Chevignon, who stay green for more time, stay green longer, as compared to other cultivars like Or grain in Sacramento in 2 different dates during the grain feeding period. So with Earth observation, we can observe some functional traits from space. So how to monitor in these 2 kind of trials with satellite on research infrastructure on farmer fields? So in farmer fields, what we consider is large plots of 40 by 40 meters, where we grow, as I said before, 6 cultivars. 2 of them are common to all sites, and the other 4 are cultivars adapted to the particular environmental conditions. We have a network of farmer fields in France, in Portugal, in Switzerland, and in Germany. I think I don't forget anyone. I think in total we have 12, if I'm not wrong, 12 different farmer fields. And in the satellite on research infrastructure, we evaluate the whole panel of 40 varieties in plots which are smaller, 80 by 80 meters. In research infrastructure, we have they have the infrastructure to grow a large number of varieties. And in the experimental design is 50 plots with 40 cultivars. 10 of them are replicated twice, and the other 30 are only replicated once, are not replicated. So this is an example of the trial in the PHENET trial in Osijek in Croatia that has been planted on October 2024. So for the satellite on farmer fields, we will rely on public high-resolution platforms. So maybe you have heard about it. Is the Sentinel-2 with a resolution of 10 meters or 20 meters, depending on the spectral bands that you work with, and a frequency between normally it's 5 days, but if you are in the right place, you can get images every 3 days. Of course, if there are no clouds, I mean, if the weather conditions are sunny, and for the satellite on research infrastructure, we cannot rely on Sentinel-2 because the resolution is not enough. So we rely on, first, the idea was to use Earth Daily, a satellite, a constellation of satellite private that were expected to be launched in 2025. So and use PlanetScope for currently as a backup. The resolution of that kind of high-resolution platforms is between 3 and 5 meters, so well adapted to the plot size of the satellite on research infrastructure, and we have more or less the multispectral bands in the visible and near-infrared that are somehow comparables to the one of Sentinel-2, but not exactly the same. So for EnviroTyping in the research infrastructure, we can rely on the persons working there. So and the infrastructure of weather stations, soil moisture, and we can expect manual observations of phenology. But in the satellite on farmer fields, we will install IoTs that will do this EnviroTyping and estimation of phenology. So these IoTs were developed last year by the society Cap Connect and also our colleagues from the unit PHACC in Clermont-Ferrand from INRAE, and especially Boris Adam, who is leading the group, that developed for us a prototype that was tested in 2024, and now we are starting to receive the operational units that will be installed during 2025 in all the satellite on farmer fields. So these IoTs, they have 2 different configurations. The full one, which includes a small weather station and RGB cameras to look at the vegetation at nadir and 45 degrees, but also watermark sensors to measure soil water potential and also soil temperature probes. And there is a smaller configuration because we will install IoTs in the 6 varieties that are planted on every site. So one of them will be full to measure also weather variables, but the other 5 will be only imaging and soil moisture and soil temperature sensor. So especially with the RGB, of course, the weather data is valuable per se, but also from the RGB cameras, we expect to validate some of the traits to not really validate, but cross-compare the traits measured by the camera with the estimations of some traits by satellite imagery, deriving some phenological dates. For instance, heading, there is an algorithm that works on daily data from a previous paper done in the group that can detect automatically heads and determine the heading dates with an error of 2 days. So these IoTs will acquire daily images and will permit us to get valuable information of the environment and the phenology of the crop. So we have plenty of observations at different scales. So the challenge is to make these observations seamless, so they are comparable across sites and across scales. So the first challenge is that in field conditions, different institutions have different devices. So at INRAE, we have PHENomobile and drones. At Zurich, they have drones and SpiderCam. And Lisbon and in OCIEC, they have UAVs. So each instrument, each vector has different instruments: LiDAR, RGB camera, multispectral camera, multi-view RGB. And every institution has developed their own algorithms to estimate some phenological traits, some traits that are relevant for high-throughput phenotyping. And these algorithms are often different. To increase the complexity, the algorithms that we use, that are used to estimate some of the traits like green area index, the FAPAR, and the chlorophyll content, are based on the same approach, but are different for the different satellites. So how can we make that these observations are comparable across instruments and sites? Okay, for the artificial intelligence algorithms. So David gave a very nice example of what artificial intelligence can do for us. Those algorithms that are based on local measurements and are validated locally, are well validated locally, they don't pose too much problem. However, we have especially some instruments and some pipelines that are available in all the institutions. So those that are transversal to all the scales and all the sites. And this is mainly the green area index and FAPAR that we can estimate from multispectral data from UAV, multispectral data from satellite at high resolution, and multispectral at, let's say, less high resolution like Sentinel-2. These pipelines need to be harmonized if we want that the green area index is comparable across sites and across experiments. How to do this? How to get seamless data sets of green area index? So let's say in the processing of data from the raw instrument data until the trait, there are different processing levels where we transform the raw data into a physical quantity, a geophysical variable that is normally corrected and normalized, and then finally a third one that is the trait. To go across these different levels. In the case of UAV, what is done mainly is an empirical calibration with reference surfaces. This is a fully empirical approach that allows us to calculate a reflectance, a canopy reflectance, from directly the raw instrument. From the satellite data, this reflectance is already provided by the image providers. And each image provider uses their own algorithms to correct the images atmospherically. They do preliminary calibrations to transform digital numbers into sensor radians. So what we get on the level 2 from these different instruments is a canopy reflectance at different bands and at different resolutions. The idea would be, okay, if we have a radiative transfer model, we apply a radiative transfer model to these different datasets, and we get a green area index for the UAV, for Earth Daily, for Planeta, for Sentinel-2, that is comparable. Theoretically, it is possible, but in practice, it's not that easy. This is the results by comparing on farmer fields, the green area index with the same algorithm estimated from Planet and Sentinel-2, and what we can appreciate is that there is a systematic bias between the sensors. These bias are due to differences in the band and their spectral response. There are, especially on satellite data, there are uncertainty in the correction of atmospheric effects. Also, we often think that the multispectral camera of the drones are robust enough, but there are a lot of radiometric uncertainties, are not as robust as the payload of the satellite, of course, and there are also environmental effects on the empirical calibration of UAV images. So the cross comparison directly from the output of a radiative transfer model is not that easy. So for that, we propose an harmonizing approach consisting, we have different gradient in resolution, which is, let's say, opposites to the radiometric quality of the images of the data provided by all the instruments. So our idea would be calibrating a radiative transfer model at the middle resolution, at 3 or 5 meter resolution, because we can get representative samples of our surface, and then calibrate and validate this model from to estimate a green area index that is robust enough. And then doing a cross-calibration exercise, including perhaps data taken from the cameras of the IoT, and this cross-calibration exercise with Sentinel-2 data, with the lower resolution sensor, needs to be done in the satellite on farmer fields, because the plot size allows a perfect rescaling of the planet data, both the planet data and the Sentinel-2 data, without the influence of possible heterogeneity associated to the plot size. So once that we get a calibrated green area index between planet and Sentinel-2, what we will do is using an empirical hybrid approach to relate this green area index with the reflectance from the UAVs calculated from a tested and validated empirical calibration method. This empirical hybrid approach can be directly through the radiative transfer model, or can be just training a machine learning method that allows to estimate green area index from the UAV reflectance. And of course, this exercise has to be done in the satellite on research infrastructure, because again, the plot size permits a perfect rescale of the planet data and the UAV data without having problems associated to heterogeneity at the plot scale. So this on what regards the intercomparability. And then if we go at the end of our use case dream, we would like to present our approach to use the models in order to estimate functional traits. So the high throughput phenotyping platforms provide us traits that are essentially morphological and structural, which are highly dependent on the environment. To characterize G cross E interactions, what we need is to estimate what we call functional traits. You have examples of these functional traits that are less dependent on the environment and are more closely linked to the genetic characteristics of the varieties. So to go from morphological structural traits to functional traits, what we poster is to use crop process-based models, because some of the morphological or structural traits are indeed canopy state variables simulated by the crop model. Some are direct state variables, like the green area index, the senescence fraction. Some others are more or less well described, like canopy height and ear density, that are not always properly well simulated by the crop process-based models. And there are others that we still need to understand how we can use them for to calibrate crop models, like, for instance, average ear size, but there are others. So the idea would be using these morphological structural traits to estimate on different environments what we call these functional traits that are essentially varietal parameters of the model that we use. For that, a colleague from us, Daniel Pasquel, who works in the national project ANR-FAST, has proposed to use in collaboration collaboration between the unit EMA and the labs, has already conducted an exercise, an in silico exercise, where he has defined a set of 80 varietal parameters of the Sirius quality model, was developed by Pierre, related to leaf growth, to light interception, to yield formation, to phenology. And then the idea is of this in silico experiment is creating what we call virtual genotypes, or sets of true varietal parameters. We simulate the observations that we can get from PHENomobile, from UAV, from satellite data. We contaminate these measurements with the expected errors that we have measured in the field for this kind of traits to create what we call pseudo-observation of structural traits that we try to use in a data simulation approach to get the estimated varietal parameters of these virtual genotypes, to answer the question of, well, how many environments do we need, how many structural traits we need to observe from high-throughput phenotyping, and whether we need some prior information about these varietal parameters. This work of on the in silico analysis of model-assisted phenotyping is still ongoing. And in this exercise, what we identify are different groups of state variables that we can observe from different scenarios of data availability. One scenario where we have only manual data, so phenology observations and descriptive measurements that we are progressively replacing by data that we can collect from high-throughput phenotyping platforms. So that allows us to measure this, to observe these traits more frequently, and also including some scenarios of availability on high-throughput phenotyping platforms, where we envisage using artificial intelligence to progressively estimate more traits that are constitutes state variables of the Sirius quality. The idea is to understand how this increase in the availability of information allows us to estimate more accurately these varietal parameters of the virtual genotypes. Of course, with the objective of repeating the same exercise with the actual data that we are observing in the phenotyping platforms and the different satellite trials. And just to finish, the last examples, some perspectives on how we can use artificial intelligence, probably to explore with our colleagues from the Work Package 5. This is an idea that a colleague from us, Christian Fournier, from the labs team had, on how, well, now from the PHENomobile, from different high-throughput phenotyping platforms, we can get valuable structural data regarding the distribution in 3D of the plant area, the distribution of the ears in the canopy, the dynamics of the canopy height, but also the plant density. There are a lot of structural information that is unexploited. The idea on this approach would be using an architectural plant model, the Adelwitt model, that could be inverted using these high-throughput phenotyping traits to estimate traits at the level of the organ. So, for instance, the average size of the flag leaves and the different leaves depending on their position in the main tiller, the number of tillers per plant, and also, well, things like, well, the ear length we can already estimate from artificial intelligence, but the peduncle length, the different length of the internodes, and this represents directly state variables of the Sirius quality model that will allow us to estimate more accurately some functional traits like the one that you see, that the potential length of the last internode, the potential surface area of leaves produced before floral initiation, and many others that can provide a deeper insight on the functioning of the different genotypes. And okay, that's everything that I wanted to present to you. Thanks a lot.